Thanks for the quick introduction about me, and thanks. I really appreciate for the Professor Murase to give me the opportunity to talk in this conference, and also I'm really appreciate for the International Research Unit of Advanced Future Studies at Kyoto University. Today, I would like to talk about some of the research about the D-amino acid in life science. We are studying the D-amino acid in the protein. Okay, in this morning session, I could see a lot of the interesting presentation for the universe. And our institute has a bottom picture, and we have the 2 research reactors. So we can use a very intense radiation for our research. So maybe I can start a new project for the protein and the irradiation. So I think it's very good for me in this morning session. Okay, now we are from the Osaka. It's next to the Kyoto. And some people are using the Kansai International Airport, maybe. And our institute is close to the airport. So maybe from the airport, you can see the 2 reactors. Anyway, today my story is about the amino acid. This picture is the backside of the drink. In the drink, in the backside of the can, there are some materials. They label the, for example, the upper panel shows I wrote the pink line. It's arginine, one of the amino acid. The bottom picture, there are the 4 amino acid. The valine, leucine, isoleucine, and threonine. But for all of the amino acid, you can see the small size L. This indicates all of the amino acid is L amino acid. Amino acid is a chiral molecule, so actually they have the 2 ways. One is L amino acid. Now you could see on previous picture, and the other one is D amino acid. It's not normal in our life, but when you synthesize the amino acid in the laboratory, you can get both amino acid 50 and 50, L and D amino acid. Both have the same structure, same chemical, same physics, but both are not the same structure in the case of the protein. So, by some reason, in our body, in our protein, there is only L amino acid. Is a component of the protein. People say this as a homochirality. Nobody knows the reason for this homochirality, but our body is made by only from the L amino acid. There are 20 amino acids in our body. I just wrote the all amino acid. The protein is a chain of the amino acid, like this one. And they can form the correct structure. They can work for the, they have the function. One of the example, we focus on the lens protein. The amino acid chain can form the correct structure, like the left panel. And this can work with the correct structure to keep the lens clearance for a long time. Back to the protein, the amino acid chain. I separate the L amino acid and the D amino acid in our body. All of the amino acid is L amino acid. Just the glycine doesn't have the chirality, but the 90 of the amino acid form is only L. So one of the big question for the life science is the reason of the homochirality. It is the same in the laboratory. In the universe, on the primitive Earth, there must be both L and the D amino acid at the same amount. But by some reason, only the L amino acid was selected to make our body. It's very interesting. But in one subject, the people found out the D amino acid from something. This is a fossil in the archaeology. In the fossil, they are out of the amino acid. Maybe this is from the they generated the protein. In such fossil, the people found out of the D-amino acid. And the increasing the D-amino acid with age of the fossil. So in this area, people can use the amount of the D-amino acid for estimating the fossil age. The fossil, it's in the under the ground, in the low temperature. So the other people think this reaction maybe occur in the high temperature, like the 37 degrees of the human body. So probably some of the amino acid is changing slowly from the L to D under the physiological condition. And that's correct. Recently, a lot of the people found out the D-amino acid in our body, especially from age-related disease tissue. The Alzheimer's disease from the brain, and from the senile plaque, people found out the D-aspartate. From the patient of the Parkinson's disease, people found out the D-aspartate residues. The aged blood vessel, aged skin, the red color indicate the presence of the D-aspartate. The cataract lens, the lens from the patient of the senile cataract, and also the age-related macular degeneration, the people have the D-aspartate. So all of the amino acid which was found in our body is the D-aspartate. So why they are out of the D-aspartate? This is just a simple picture. I showed the out of the amino acid chain as a protein. There are many aspartate in the protein, but with aging, only for the specific site-specific, the aspartates is changing from the normal L-alpha aspartate to the D-aspartate. During this chemical reaction, they also change to the beta aspartate. So in our body, in aged tissue, there are the three abnormal aspartate. The next is a scheme for this reaction. This is completely the chemical, non-enzymatic, but spontaneous reaction in our body. So from L-alpha aspartate, they process to the 5-ring 16-meter intermediate at the center of the picture, and they change to the L-beta, D-alpha, D-beta aspartate in our protein. So what's going after the formation of the abnormal aspartate? From L to D, the side chain direct to the opposite side. In the physiological condition, the side chain of the aspartate have the negative charge. This means a negative charge, direction of the negative charge in the protein direct to the different side. This means they alter the structure of the protein. They alter the interaction between the protein. From the alpha aspartate to the beta aspartate, they extend the polypeptide, they extend the chain of the protein. The increasing the C number means increasing the hydrophobicity of the molecule. This also impacts on the structure of the protein. Also impact on the stability of the protein. So D-beta aspartate has both. So presumably this is the most toxic for the protein. Okay, by some reason, by the reason of the homochirality, all of the protein have the correct structure made by only the L amino acid. If aspartate is changing from normal L to D-aspartate, they form the abnormal structure. They interact each other. They form the protein aggregation like this one. And this is precipitated in the aged tissue. This may cause age-related disease. Especially people say the protein aggregation disease, such as the Alzheimer's disease, the cataract, and any other disease. I'm studying about the lens and the senile cataract formation with aging. This picture, the people has right eyes is a correct lens. The left eyes has a cataract. It's a little bit whitey. So when we was young, our lens has the correct structure and the correct function to keep the lens clearance. So we can see the very beautiful picture. But with aging, it's a little bit whitey. At the 50 years old, the picture is a little bit whitey. At the 80 years old, these people cannot see anything. We cannot escape from the cataract. About the 40 to 50% of the people who are 50 years old has the cataract. And over the 80 years old, almost 100% people has the cataract. When you go to the outside, and if you feel something is very shiny, it's already starts the cataract. This is a simple model for the formation of the cataract. The normal L amino acid forms the correct structure of the lens protein. They can keep the clearance of the lens. Once they form the abnormal amino acid in the protein, they form the aggregate precipitated in the lens. Inhibit the sight of the from the eye, and you cannot see anything. So it's very important reaction, but so far nobody knows. There is a reason. So this is the reason about the why no one knows. Usually, when we analyze something, when we check the correct structure of the protein, correct structure of the amino acid, we are using the mass of the amino acid. But the alteration from the L amino acid to the D amino acid doesn't change the molar mass. By this reason, when we check everything, we couldn't find out the abnormal amino acid from the protein. And to identify the abnormal D amino acid, in my case D aspartate, we developed the method to identify the D aspartate. To do that, we are using the mass spectrometry. This is a brief introduction of the mass spectrometry. We are using the tissue for this instrument. In the PC, they can calculate, they can get a lot of the data. They automatically analyze for each amino acid, and they can give the result. Usually, we just check the result, but we don't know what's going on the PC. So to identify the abnormal, the same molar mass, the D aspartate, we change the method for this instrument. It is very simple, different from the original one. We just calculate the abnormal amino acid, and ask the PC to whether the present or not present the same molar mass with different something. This is the normal mass spectrometry for the 83 years old lens. In the 83 years old lens, there are a lot of the protein fragment. And we ask only one mass from a lot of the data. In theory, and in original usage, they show only one peak, like the bottom picture. But it may be wrong. There must be something in the data set. And we use our method to do that. And we found out 4 independent peaks from the one mass. Our hypothesis is true. There is not a single normal aspartate in aged tissue. Now, we could see the 4 peaks, A, B, C, D. And we don't know now which one is D aspartate. And also which one is beta aspartate. This is a method what we are using now. After getting the multiple peak from the aged tissue, we added the enzyme, 3 different enzymes. One enzyme is the digest for the normal L-alpha aspartate. The peak shows the mass. Once they digest to the 2, the mass is a different one, different 2. So when we choose the one mass on the system, the peak is deleted as a right top panel. Now we can see which peak is from the normal L-alpha aspartate. The second enzyme, which add the methyl residue for the amino acid, L-beta aspartate. After modify the peptide, after molecule the amino acid, they change the molar mass, increasing the 15 Dalton. This is the same as the digestion. They are going out from the one mass, they show the different mass. I mean the peak is deleted at the B. Now we can see which one is from the L-beta aspartate. And finally, we are using the D-alpha aspartate digestion. It's the same as the L-alpha aspartate digestion. From this data, we can say the C is from the D-alpha aspartate. Now there are no enzyme to identify the D-beta aspartate, but when you see the three picture, there is only one peak which was not digested, which was not modified from any other enzyme, D. Now we can say D is from the D-beta aspartate. I use this method to identify. Now we are using this method to identify the abnormal aspartate. And we can see the L-alpha, L-beta, D-alpha, D-beta aspartate from the aged tissue. Now, we need to check the ratio between the L and the D, and also the age of the human. So we get the out of the eyeball. We use the oral eyeball for this assay. Compare the each age dependent from the each age. The merit for our method is very fast. In this assay, we can do everything only for the 2 days. But if we choose a classic method, like the way for the fossil, we needed 100 days. So with our method, we can use this method for the out of the eyeball and to get the out of the data to compare each age. And this is the result. So red line shows the normal L-alpha aspartate at the position of the blue ball in that structure. This is a lens protein. On this aspartate, it is changing from L-alpha aspartate, red line, to the blue line. Blue line is increasing with aging. Blue is a D-beta aspartate. Like this way, it's very clear. They have the age dependence. So until recently, we got the method to detect the D-aspartate in the aged tissue, like this one. The lens in the eye, one of the aspartate is changing from the L-alpha aspartate to the C abnormal aspartate. That's good to know, but we actually don't know after forming such kind of aspartate, what those aspartate contribute for the lens function. It's very important to know this one, because we want to check the contribution for the cataract formation. One of the big issues is there is no way to make the mimic protein like this one. It has been impossible to pinpoint insert the abnormal aspartate, abnormal amino acid in the protein. So we try to develop the brand new method to be possible to make this one. Yes. And, I do not talk a lot about this one, but we use this. I just want to quickly introduce our method. The white alphabet indicates the presence of the amino acid chain. I made all of them, most of all part in the cells. On the other hand, the blue line shows a chemically synthetic peptide. I synthesize the peptide, and I replace the aspartate in one place to the other aspartate, and chemically ligated the both to make the protein. By this way, we could make some kind of the protein which containing the pinpoint abnormal aspartate. And I also show the decreasing the function ability of the protein. Anyway, I just compare only the L aspartate to the D aspartate. as people thought the L-aspartate is decreasing with increasing the D-aspartate even in our body. And they decrease the function of the tissue. And they may cause age-related disease in our body. The model of the cataract. The normal protein has only the L amino acid. With aging, the site-specific aspartate is changing to the D. They start to aggregate to induce the finally cataract formation. So if we can suppress the chemical modification from L-aspartate to the D-aspartate, we can prevent the cataract formation. So far, once you have the cataract, you can have the surgery. But it's because you are living in the developed country. But in many countries, you cannot have the cataract surgery. In such nation, you need the very simple prevention for the cataract. So we want to suggest eye drop to prevent chemical modification of the aspartate to suppress the cataract formation. The important thing is that this is a suppression of the chemical reaction from L to D. So you can use this medicine for everything in your body. So if you can get this eye drop, you can use this for any part of the aged tissue. So probably it's possible to prevent any of the age-related disease. This is made by the D-aspartate formation, and it prevents the aging to keep the quality of life in the later phase of your life. So I already talked about the origin of life. In the primitive Earth, there is only the gas, radiation, and they form some amino acid. Should be both L and the D. But by some reason, only the L amino acid is selected. Nobody knows this reason, so I want to try to search the reason for this event. And also, the stories are a little bit different in the space. In the space, like in the contents of the comets, L amino acid and the D amino acid is almost 50 and 50. It's very close to the primitive Earth. And now a lot of the people, a lot of the group could get a lot of something from the space. After they gave back to our Earth, maybe we can see the ratio between the L and the D from their something. And this is a last slide of my favorite picture. There is a picture from through the looking glass and what Alice found there. This is very popular. The Alice is living in our world. Our world is made by the L amino acid. But she goes to the different world through the mirror. Once she went to the mirror, in that world, there is everything the opposite way, like the D amino acid and the L amino acid. In this world, Alice drank the milk. But milk was made by the D amino acid. She said it's not so delicious. That's true. It's completely different one. Maybe the milk has a very old taste. So now we are living in the Earth, and we can see a lot of the life. Everything was made by the L amino acid. But in other planet, if they select the D amino acid at the origin of the life, everything should be made by the D amino acid. So in the universe, in the other planet, there may be the life who made by only the D amino acid. So today I talked from the origin of the life to the D amino acid is increasing during the aging. But it's very interestingly, the origin of life is a very big event for the life. But our life is only maybe 100 years. So somebody decides homochirality. But such homochirality is break down only in our 100 years old life. So I have very interesting for this D-amino acid research in the from the primitive Earth to the people aging and also the space in universe. So thanks for your attention.